And so let me end on a description of interacting with the CHOP Sequencing and Analytical Center. So interest in the microbiome has been soaring. Here I searched on PubMed on the term microbiome as a function of year, and you can see the sort of exponential growth in papers on the microbiome. I and several folks in this room have been on multiple NIH panels on how to foster microbiome research within each of the different institutes. It's great cocktail party conversation. I mean, fecal transplants, everybody wants to hear about that. The Human Microbiome Project was the largest project in the history of microbiology, and Penn and CHOP labs were leading participants, so we're well positioned to go forward in this area. The leadership at Penn and CHOP have been pretty clear that they want to see the microbiome research here kind of focused on improving patient welfare. So many folks are focusing on animal models or human association studies, looking for correlations. What we're hoping to do is get much more into the human intervention studies. I mentioned the PLEASE study, the pediatric study, for example. And we're trying to set up and develop tools for doing so. Multi-omics integration, and another focus is trying to be lifelong, merging pediatric, adult, and old age cohorts in, within the microbiome program. So this just shows the overview. We report to leadership at CHOP and Penn. Bob and Gary are here, 3 directors. We have an internal advisory board, including Joe here at CHOP and several other CHOP folks. We have an administrative core, sequencing and analytical core, which is what this lecture has been about. But several other cores as well, one for microbial culture and increasingly microbial metabolomics. We also have a notobiotic mouse facility led by Youngwon Choi. That is germ-free mice where people can introduce new bugs or ask what features change in the mouse phenotype in the absence of microbiota. And then a couple of cores to help with human interventions. Jim Lewis is helping with study design and Ron Coleman biobanking for acute care specimens. So the CHOP Microbiome Center is now located at a number of different sites. The nucleic acid biochemistry sequencing is led by Dr. Ravi with Lisa, Casey, and Dorothy, shown here. Their site is here in Abramson. The sequencing center itself is in the basement of Colket next to BGI, the Beijing Genomics Institute. And Kyle Bittinger is leading the analytical center at 3535 Market with Ashwini Patel and Shailan Taines. So we're a little spread out, but a lot of folks now who are trained up and ready to go. The sequencing center, the technicians are smiling because the instruments are now starting to run. We have 2 HiSeq instruments, a MiSeq and a Pacific Bioscience sequencing instrument. The last one being useful for bacterial whole genome sequence data acquisition. What I've mostly been speaking about is work with HiSeq or MiSeq or the older 454 platform. So we have a website that's useful for interacting with the center. It's just gone live in the last couple of days. So this is the address, but if you want to find it, search on PennCHOP, one word, and microbiome, and that'll take you to the website where you can get a bunch of information about what we're up to. So it's built by April Weekly and Laurie Zimmerman. So just to show you how to propose projects and get started working with the center, if you click on resources, you can see the CHOP Microbiome Center. If you go to the top page, click on this, it takes you to the CHOP Microbiome Center, sequencing and analytical resource. And if you scroll down on this page, there's Kyle again, you'll see operational information. Please fill out this form and send to Jesse Ehrlichman. And so this is, you can fill this out and send it to us as a way of sort of getting in the queue and initiating a conversation about starting your project. And so a little more about that. We have several formats for interacting. One is sort of initial consultation or just brainstorming, or can you give me some suggestions for starting a microbiome study? Another is straight fee-for-service. We're going to, if someone has samples or libraries or what have you, they just want them worked up, sequenced, send back the sequence. Center takes no responsibility for the outcome. That's fine. We're happy to do that sort of fee-for-service analysis. And then the third format is going to be collaborative, where the customer works closely with the center, brings a sample set, and folks at the center help figure out the statistical framework, maybe power analysis, the association of the analysis and the questions posed. Then samples come to the center, their DNA is prepared, libraries prepared, sequencing. Data is then analyzed at the center, and a series of predetermined analyses carried out and sent back to the customer. And so this is a great format. We want to be real careful up front to make sure people's expectations are aligned with what they're going to get at the end. It's very common that with scientists, when you see some data, you'll say, well, what about this? What about that? You'll want to do some follow-up. And that's fine. The center's going to support that, but that'll be sort of additional fee-for-service analysis. We want to be kind of careful to have a finishing point and then, add on further stuff at the end. Also, it's really important, I think, that we be involved in the design up front if we're going to do the analysis at the end. I can tell you from personal experience that it's sort of demoralizing and useless to have a sample, a study that's designed in a way that can never be successfully analyzed, and sort of is demoralizing for all involved. So I think it's really important and hopefully helpful for the center people to be involved right at the beginning in order to help with design, so there's a good study that can be completed at the end. And so here on this form, we'll ask you a bunch of stuff: your name, your how you want to work with us. Do you have IRB approval? And another thing we'd like is a paragraph or a few sentences at least on what the project is, what the questions are, what the sample set is, what the relationship between question and data is. Because what we do is take that paragraph and put it at the beginning of the report at the end. So we keep the data figures, the output together with a description of its importance, so that you can come back years later and see what the analysis was, what the point of it was. And again, we found from experience in other areas that it really helps to have a clear description that can then be stamped on all the outputs, so there's no question about it when you come back years later. So a couple last points. We're hoping to have calls for pilot grants this fall. So we're hoping to have funding going out in the community to help use these resources and do these kinds of studies. And we'll be having a microbiome symposium here at Penn and CHOP, October 28th and 29th. So a chance to hear some of the big noises in the field and learn more about it then. And again, to work with us, to follow up, go to the website, contact Jesse, as shown here, fill out the form and send that back to us, and then we'll put you in the queue to meet with staff and design your study. Okay. So just to reemphasize, microbes come in communities. What those communities do matter to human health. So bring us your samples. We're ready to go. And so as a last slide, I'll just show all the folks at Penn and CHOP who've collaborated with us and formed the microbiome group here. It's really quite extensive. We really are one of the best places on the planet for doing this kind of research, and we'd like to invite all of you to get involved if you want to. So I'll stop there and take any questions.